Good morning, students. Today we studied about structural changes in chromosomes. Variation in the structure and number of chromosomes have been observed in natural populations and could also be produced artificially in a variety of organisms. These variations have been extensively studied and are described as structural changes and numerical changes in chromosomes. Structural changes in chromosomes and the numerical changes in chromosomes. Structural changes in chromosomes can be of following type. First is deficiency, which involves loss of a part of chromosomes. Second is duplication, which involves addition of a part of chromosomes. Third is inversion, which involves a reverse order of the genes in a part of chromosome. And the fourth is translocation, which involves exchange of segments between non-homologous chromosomes. These structural changes are diagrammatically represented in this diagram, where two non-homologous chromosomes from the complete set are shown. Structural abnormalities may be found in both the homologous chromosomes of a pair or in only one of them. When both homologous chromosomes take part in these changes, these are called structural homozygotes. Deficiency, homozygote, duplication, homozygote, etc. If only one chromosome takes part, this will be called structural heterozygote. The constitutions of a translocation heterozygote and that of a translocation homozygote are shown in the second diagram. Structure here we can see in the diagram two non-homologous chromosomes. There is a deletion and addition. In first we will see the deletion of part third. In next addition there is a addition of another extra part, and in duplication, translocation, exchange between two chromosomes. Different kinds of changes in chromosome structure. Deficiency is due to loss of a part of chromosome. Smaller deficiency present in heterozygous conditions can be tolerated by an organism. Such individuals at meiosis will form a loop in a bivalent that can be observed at Pachydian stage. Loops can also be observed in salivary gland chromosomes of Drosophila, which are found permanent state of pairing, so that even small deficiency could be detected in these chromosomes. Deficiency have an effect on inheritance also. In presence of a deficiency, a recessive allele will behave like a dominant allele, which is called pseudodominance. This principle of pseudodominance exhibited by deficiency heterozygotes has been utilized for location of genes on specific chromosomes in Drosophila, maize, and other organisms. L. J. Stadler, who was a pioneer in the radiation work in plants, devised a method where a homozygous recessive stalk was pollinated by irradiated pollen from dominant stalk, so that if radiation induced a deletion involving the gene concerned, recessive allele will expressive due to pseudodominance. In this figure, If homozygote ABC pollinated by ABC heterozygous, a recessive parent with a dominant parent, F1 will be produced expressing only dominant characters. If pollen with dominant alleles is irradiated, a deletion may be induced, leading to expression of pseudodominance by one or more recessive alleles. If meiosis at Pectitin is examined in such a deficiency heterozygote, presence of loop will be indicate location of genes. Several genes were located on different chromosomes of maize and tomato. Utilizing such deficiencies in Drosophila, also deficiencies were recorded, particularly on X chromosomes in regions of W, white eye, facet eye, and vermilion colored eye. Deficiencies have also been recorded in Walsing mice in region of gene V, inducing nervous abnormality. In human beings, a deficiency was discovered which was associated with cat-like cry, so that the child carrying this deficiency had a cat-like cry and also had microcephaly, small head and low mental faculty. This deficiency was found in a segment of the fifth chromosome. So, we discussed about the deficiencies in the chromosomes, chromosomal structures, which involves loss of a part of a chromosome. So, it will show the structural changes in chromosomes by deficiency. It's completed. Thank you.